Good morning, students. Now we have reached towards the end of the chapter, and today we are going to discuss about the different organelles of the cell. Now we can define organelle as the specialized membrane bound structures in the cell concerned with the definite functions. Different types of organelles and their main characteristics as well as the chief functions we will see one by one. So let's start with endoplasmic reticulum, which in short is termed as ER. Endoplasmic reticulum is also called as the skeleton of the cell. Okay, and you can see an enlarged view of endoplasmic reticulum here, and uh, now come to the main characteristics of endoplasmic reticulum. So endoplasmic reticulum is irregular network of double membrane tubules distributed over entire cytoplasm of the cell. Means a major portion of in the cytoplasm of the cell uh, is being occupied by the endoplasmic reticulum in tubular form. You can see here it is present in the tubular form like this, and it remains connected with the cell membrane at its inner end. You can see here, and towards the outer side it remains connected with the plasma membrane. This is just a symbolic diagram, so here it is not being shown, but towards its outer end it remains connected with the plasma membrane of the cell. Next very important thing about endoplasmic reticulum is that it is of two types. If on endoplasmic reticulum ribosomes are present, you can see small granular structure here, which is being represented in pink color. So these are ribosomes. If ribosomes are present on the surface of endoplasmic reticulum, then it will be called as rough endoplasmic reticulum. When you will observe this endoplasmic reticulum under microscope, you'll find its surface very rough. You'll observe very rough surface over it, over here. So it is called as rough endoplasmic reticulum. And if the ribosomes are not present on the endoplasmic reticulum, it will appear smooth. So it will be called as smooth endoplasmic reticulum. Now let's move towards its functions. So endoplasmic reticulum provides a framework of the cell, isn't it? That is why it is called as the skeleton of the cell also, and it provides the strength to the cell. And next important function is that rough endoplasmic reticulum, because ribosomes are present over there, so they help in the synthesis of protein. RER provides site for synthesis of protein. Protein synthesis is the work of ribosomes. Next is the smooth endoplasmic reticulum. Both kind of endoplasmic reticulums are present inside the cell. Smooth endoplasmic reticulum helps in the synthesis of fat molecules or lipids, we can say. Endoplasmic reticulum serves as a transport system of protein and fats, because it remains distributed over a very large portion of cytoplasm, remains connected with the nuclear membrane at one end and with the plasma membrane at the other end. So it provides a passage for transport of materials inside the cell. Some of the proteins and lipids which are synthesized by these two types of endoplasmic reticulum uh, are utilized in building the cell membrane, and this process is called as biogenesis. Another very important thing is that in liver cells of vertebrates, smooth endoplasmic reticulum especially plays a crucial role in detoxifying many poisons and drugs. Next come to the Golgi apparatus. Golgi apparatus is also called as the delivery system of the cell. Can you see here the structure of Golgi apparatus? It has stacks of flattened membrane sacs called cisterns. These are called as cisterns, and along with these vacuoles are present and vesicles are present. Vesicles are small bag-like structure, and they are generally found near the nucleus of the cell. And this Golgi apparatus, because was discovered by Camillo Golgi, that is why it name has been given as Golgi apparatus, or sometimes it is also called as Golgi body. Now come to its functions. They are concerned with the secretion of the cell, including the enzymes, hormones, etc. And as well, they help in the formation of acrosome of the sperm. Do you remember structure of sperm? You have read it in standard 8. At the top of the sperm, uh, one oval structure is present, that is called as acrosome, which plays very important role in fertilization by dissolving the membrane of the ovum. As the membrane of the ovum gets dissolved, nucleus of the sperm gets its entry inside the ovum, so that the union of the male nucleus as well as the female nucleus can take place. So acrosome is a very important structure which comes from the Golgi body. Next, they help in the formation of lysosomes also. Lysosome again is an organelle, and we'll discuss it later on about it. Next, the function includes the storage, modification, and packaging of different things in its vesicles. Now come to lysosomes. Lysosomes are called as suicide bag of the cell or waste disposal system of the cell, or sometimes called as defense organelle also. Can you see structure of lysosome? It is again a bag-like structure, membranous bag-like structure, which has been made from Golgi body itself, as we have discussed right now. It contains almost 40 types of digestive enzymes, which are called as hydrolase. And these hydrolase are responsible for dissolving or destroying different things, which we will discuss right now. Digestive enzymes of lysosomes are made by endoplasmic reticulum. Now come to its functions: intracellular digestion, then lysosomes destroy foreign substance, it destroy the destroy the bacteria which has been entered in the cell, and that's why it is called as defense organelle. Sometimes what happens, if metabolic activities of the cell has been damaged, then that damaged cell is destroyed by its lysosome itself, and that is why it is called as suicide bag of the cell also. Next comes the mitochondria. Mitochondria is also called as powerhouse of the cell, and we have already read a lot about it. Mitochondria is a double walled structure. Outer membrane is porous in nature, and an inner membrane is deeply folded. Can you see these projections? These projections are in its inner membrane, and these projections are called as folds are called as known as cristae. And role of cristae is to increase surface area for providing the site of respiration, to provide bigger site for respiration. Next is that they have their own DNA and their own ribosomes itself, so they can make their own protein. Now, as far as the function is concerned, we all know that mitochondria provides site for aerobic respiration, and respiration release of energy from pyruvic acid produced in the cytoplasm, and then the same energy gets stored in the form of ATP. ATP stands for adenosine triphosphate, and this ATP is called as energy currency of the cell, and which is being used for various metabolic activities and functions of the cell. That is the reason why mitochondria is called as powerhouse of the cell. And the mitochondria also synthesize respiratory enzymes which helps in respiration. Next comes the plastids. Plastids are present only in the plant cell. Now can you see here the structure of plastid? Again, it appears almost similar to the mitochondria. It is also double-walled and contains its own DNA and ribosomes. Plastids can be of different types. The types and functions are as follows. The first one is the chromoplasts, which contains the colorful pigments. And one thing which is very much important you should keep in your mind is that plastids are present only in the plant cell, not in the animal cell. So this chromoplasts contains the colorful pigments, and the role of chromoplast is to impart color to flower, fruits, and etc. Leucoplasts are those plastids which does not contain any pigment, and so they are colorless, and their role is to store starch, oil, protein, granules, etc. Cells of potatoes have leucoplasts. Next comes the very important type of plastid, and it is chloroplast. Chloroplast contains green pigment called chlorophyll, and we should not understand that no other pigment is present over there in the chloroplast. Other pigments are also present in the chloroplast, like orange, yellow, but what happens? The green pigments are much more as compared to the yellow or orange pigments, and that is why they appear green in color, but it doesn't mean that it has only green pigments. And we all know that chlorophyll is very essential for carrying out the process of photosynthesis, because the very first step of photosynthesis, that is the trapping of solar energy, is possible just because of this chlorophyll. Now come to the next organelle, that is vacuole, and vacuole is the non-living structure again present in the plant cell. Okay, and vacuole you can see here again is a sac-like structure, a bag-like structure, and in mature plant cell, single large centrally located vacuole is present. Animal cells, it may remain present, may not remain present. If present, they are very small and temporary. And they act as storage sacs containing water or other substances in the solution form, and covered by a membrane which is called as tonoplast. It means the membrane of vacuole. The outer layer of the vacuole is called as tonoplast. And the very interesting thing is that almost 50 to 90% of the cell volume in plant cell is occupied by this vacuole itself. And fluid which is present inside the vacuole is called as cell sap. Now come to the important functions, as we have discussed right now: storage of water, other substances, food, pigments, and waste products. Then it gives turgidity to the plant cells by pressing against cell wall. I hope you can understand this term turgidity here. Next, stores important substances like amino acids, sugars, organic acids, and some proteins as well. In amoeba, food vacuole contains food items that amoeba has consumed. And in some other unicellular organisms, vacuole is being used or held in expulsion of the excess water present. Now we have reached to the stage where we can differentiate between plant cell as well as animal cell. So one by one we can see here size. In plant cell they are larger, animal cell they are usually smaller as compared to plant cell. The cell wall is made up of cellulose here. Here no cell wall is present. Cytoplasm is not so dense, but denser cytoplasm is present and which are granular in nature. Then plastids are present here in plant cell. No plastids are present here. Vacuoles are large and prominent. Vacuoles, if present, they are very small in size and temporary in nature. Nucleus is located close to the periphery, and here nucleus is almost centrally located. And one very important structure, centrosome, which helps in cell division, is absent in plant cell, but it is present in the animal cell and initiates and regulates the cell division. Now, the very last topic of this chapter is cell division. Actually, my book is of old edition, but I hope yours is of new edition. And in new edition, one more topic has been added, cell division. This is just the introductory topic, and we'll learn just only about the two types of the mitosis in brief, two types of the cell division in brief. The first type of cell division is mitosis, and it occurs for growth and repair, and it occurs in the somatic cells, that is the general body cells. It continues throughout the life. Here what happens, one parent cell divides into two identical daughter cell, and full set of chromosomes is passed on to each daughter cell. In case of human being, the full number of chromosome is 46, and this is called as diploid number of chromosomes. So here with the diagram you can understand the parent cell divides into two daughter cells, and you can see 2n means 46 chromosomes are present here. 46 chromosomes are present in each of the daughter cell also. The next is type of cell division is meiosis, and it occurs only in the reproductive cells, and does not continue throughout the life, but only in the reproductively active age. And one parent cell here divides into four daughter cells. You can see in this diagram the parent cell first division gives rise to two cells, and then each of the cell further divides into two, and at last we get four daughter cells from the single parent cell. And here a very important thing is that only half number of the chromosome is passed on to each daughter cell, and this is the haploid number of chromosome, means 23 chromosome is present in each of the daughter cell and not the 46. So this is how we come to the end of the chapter. Go through the notes carefully, try to understand, and solve the assignment which is given here. Okay, now till the next video, goodbye.